this, and this, and whatever that is
Hey,
going
it's
to go away.
recording.
Yeah. All right, so we should be good. Is that full screen? Pretty
Yeah,
close.
that's good enough.
Okay. Yeah.
All right. So yeah, we were saying, so these receptors are on other cells. There's things there. These that the CAR T cell is binding to are receptors or kind of things that are sticking out from other cells. So they're attached to other cells. They're strengthening that attachment. So they're kind of just holding hands with another cell and things like that. And then these are just some markers that they're looking for in specific types of cancers and some other targets. This is the actual region of the CAR T cell that they can put in. These are the things they're looking for, and they can put the antigen for those at the top of the CAR T cell here so that it binds to that region. These are just some examples. And so let's again talk about the kind of makeup of the actual CAR receptor versus what it was modeled after, the T cell receptor or TCR. So don't worry about all of these things. Just to point out, 41BB is here. So this is in normal T cells or cells. 41BB is not directly attached to the TCR, but it is associated with the signaling cascades that they produce. And so that is one of the intracellular domains of the CAR T cell, of the CAR receptor makeup. Another region is CD3 zeta domain. That just means zeta. That's an intracellular portion of the actual TCR that's responsible for much of its actual signal transduction. And then another co-stimulatory molecule that's close by is CD28. So that's what it looks like in an actual cell. But when we put these when we make these in the lab and then put them and express them in patients' T cells, these are more what they look like. So we're kind of taking all of these things, we're throwing them in there. So they work pretty similar actually, but they look a little bit different, and this is obviously a kind of dumbed down version. It's important to notice that they aren't actually sticking out of the cell anymore. So it's just the catalytic portion that's responsible for signal transduction that are being expressed in these regions within the CAR T cell. So let's talk a little bit more about some of the receptors, or at least the signal transduction part of the receptors that are used to make up the CAR. CD28. I know you guys probably talked a lot about tyrosine kinase receptors. CD28 is what's known as a non-tyrosine receptor, or a receptor non-tyrosine. Same thing. So it's very similar in the signals they transduce, but there's some differences that we'll talk about. And this is a co-stimulatory molecule for the TCR generally. So it's these when the TCR gets activated, these receptors all work together to try and strengthen that signal. And these are just kind of schematics of what it looks like in actual cells. Whereas within the CAR, we only have really this portion again that's being expressed. Another one is the CD3 zeta domain. It is also a non-tyrosine receptor. And it's, again, part of the intracellular region, one of the many intracellular domains of the TCR. And it plays a big role in its signal transduction. So it makes sense why they include it. And another one is part of the tumor necrosis factor family, something I think your guys' book talks about, but maybe that don't focus on in class. And it's 41BB. And again, it's binding to antigens on other cells. One of the newer domains that they're, the original schematic only showed 3, but that's just 3 of the ones that I thought were applicable to the pathways we're going to talk about. They're also the most common, but one of the newer ones they're adding to it is this IL-2R beta domain. So it's just IL-2 receptor beta is what that means. So usually IL-2 is a cytokine that can kind of bind, or interleukin that can bind to this receptor. It is a cytokine. This is also a non-tyrosine receptor, but it's a different signaling cascade than the other ones. So I thought it was good to mention. And it's starting to be used in a lot of the newer generations, so and it kind of has some cool capabilities. So let's talk a little bit more about the actual signal it produces. This is from your guys' book. And this receptor follows the JAK-STAT signaling pathway. And so it's non-tyrosine receptor because these JAK kinases associate with these polypeptide chain, or these amino acid chains, but they're not actually, as in a tyrosine kinase receptor, they're not actually part of that amino acid chain. So this receptor itself doesn't have catalytic activity. It only has kinase abilities because of those JAK proteins that are on it. And so your book talks about this more. This pathway results in the dimerization of STAT, which can translocate to the nucleus and act as a transcription factor. Some of the things it does is it aids in cytokine release and important for chemotaxis and attraction. Talk more about the transcription factor side of things when I come next time, but this is kind of useful. I thought it was applicable to some of the stuff you guys, it's covered in your guys' textbook. Yeah. This is kind of messy. Don't get overwhelmed. The important part here is it's actually very similar, and this is very blurry, which I didn't realize it was going to be, but this is also from your textbook. This is the 4-1BB domain. It's the tumor necrosis factor receptor family. A big thing that this family is signaling cascade is associated or shown to do is it results in the free NF-kappa beta production. And it does this through the signaling cascade, which actually activates a destruction complex of IKK subunits, and this allows for the phosphorylation of IK beta to be phosphorylated, which actually marks it for destruction in this case, which I know is kind of a weird way. Usually phosphorylation, we think of activating things, or dephosphorylation, turning things off, but here this allows it to be ubiquitinated or marked for destruction, which frees up NF-kappa beta to kind of translocate and enter the nucleus and affect gene expression and stuff. And so, there's other things going on here too, which we don't need to focus on at all. But it's kind of interesting that this is the same thing that's happening in a normal cell, and we're able to get there through only using the intracellular, signaling portion of it. Kind of a way to hijack and modulate these pathways. Another one is the CD28 non-tyrosine kinase receptor. And again, all of these names here aren't important. I'm sorry, it's so blurry. It didn't look blurry on my computer. But what is important here is, again, the difference between tyrosine kinase receptors and non-tyrosine kinase. This autophosphorylation only occurs in tyrosine kinase receptors, as non-tyrosine kinase receptors lack that self-catalytic ability. Here, they rely on an enzyme called LIC, which is just a kinase, to phosphorylate on a tyrosine residue that kind of gets this pathway kicked off and going. We'll dive a little bit more into this, but there's some regions that are very prevalent to some of the stuff you guys are talking about in class. And it's just getting there from a different way. Like, I hope this isn't confusing you guys that, tyrosine kinase receptors, like, growth factor receptors, like VEGF and PDGF, those kind of activate similar pathways, but it's just a slightly different way. And so it's kind of important here that we're activating actually multiple pathways. So I know you guys talked a ton about RAS, that's being activated through this pathway. Actually, I know it looks a little different than what it looks like in your book, but it's actually very similar. This is just a different way of kind of drawing it out. It ends up resulting in a very similar result, in terms of transcription factor activation. It also activates the mTOR pathway, which talked a little bit about in your book. And again, this NF-kappa beta pathway, which is activated by also the tumor necrosis factor, but it's activated in a different way here, which isn't super important, but it is interesting, right, that we can activate these pathways that are similar or the same in different ways. That's kind of an important idea in biology. This is actually from your book as well. The mTOR pathway is super important for cell proliferation, and I'll talk a little bit more about the transcription factors it produces, or induces the expression of. And it is really cool, I think, that they can put these domains into a CAR T cell, and they still follow very similar signaling cascades. It just kind of shows how malleable all of these receptors and signaling transduction region or initiation regions are within a cell. Because the main thing to remember here is that this cell or this receptor that we're expressing is within actual normal T cells. So all of this, everything else still works the same, and we were just able to modulate it by what we're expressing and what we're having it bind to. And I think that's really cool, and it kind of shows the power of and the difference in treating disease here. So some of the key takeaways, I think, that the receptor is made up of different regions, and that these regions are generally non-tyrosine receptors and tumor necrosis factors. These regions are triggered together kind of simultaneously through conformational changes when the ligand binds to another marker or antibody on another cell. So they're expressed together, which is still very similar to how they're expressed and turned on in normal T cell receptors. A little bit different, but ends up with the same result. And then each of their signaling cascades is still the same as in the normal cells, which I've kind of beaten home. And this idea of parallel pathways, I think, is really crucial to biology, and it's something that keeps coming up. And it's all of these cascades are kind of culminating and pushing the cell in a certain direction that we'll talk about more next time. But it just kind of, the signals are overlapping, they're expressing together, and that's because the CAR T cell is trying to push the T cell in a certain direction to drive a response to help, defeat and kill cancer cells. And these parallel pathways are super important for that, because we're allowed to amplify these signals. It creates flexibility, and it provides redundancy. And these parallel pathways are also super important in just understanding, molecular and cellular biology as a whole. Any questions?
So are you physically building the receptor?
Am I?
Or cell therapy, is it physically building the receptor? And then if that's the case, how do you get the receptor to bind to the cell?
So I think I talked about this last time, which was a while ago, but these T cells are isolated. I should have an overview about it again, but they're isolated from patients, and then their genome are edited through various mechanisms, some involving CRISPR, some involving retroviral transfection. And then those are expressed by the actual cell's own machinery. And so it's built from, you're kind of inducing it, and then you're building it from within the actual.
Okay. And my kind of follow-up question to that is the medicine that you give the ligand that activates these, or is the ligand coming from other cells?
It's definitely from other cells that have, you're targeting the cells, looking for specific targets. Let me go back. So BCMA is expressed highly on malignant multiple myeloma cancer cells. So that's the target you're looking for. You don't have to give anything extra, really. You just know that your target is going to be highly expressed on the cells you're trying to kill. That's the idea, at least. It's not perfect, though, always.
You had mentioned the first three receptors that you went through, like CD28 and those guys, and then saying that those had a similar signaling cascade compared to IL-2B NRT or NTR. What is the big difference between those types of signaling cascades?
Between CD28 and IL-2R?
Yeah, or IL-2B. You said that one was different than the others.
Oh, yeah, so what I was saying is that it's different. It's a non-tyrosine receptor, which CD28 also is, but it's different in the sense that it uses this JAK-STAT pathway, which is just a well-known pathway, and it's just a different way than CD28, where CD28 relies on this LCK kinase, which isn't super important, but it's just a different way. But in this, transcription factors are different, but they're still both non-tyrosine receptors, just different subtypes. Cool.
Is that for now? I'm just gonna clap. Yeah, 'cause you're doing that's really hard to say that stuff with that much clarity. So thank you, Riley. Yeah, that was really well done. And
I know you guys have a test coming up too, so maybe some of the other pathways.
It's the test. Right, Riley's writing the test, you guys. Get over it. Deal with that. And yeah, thank you.